LOL. I'm just playing with this. Don't worry. Come on, Okay, I'm not gonna keep playing with this. This is gonna make it for a fun recording in the beginning. Um, don't worry, I got it. I got it. Beautiful. There we go. All right, so how about we get started? Oops, that's not great. There we go. All right. So today, I get to talk to you about something that's very dear to my heart. Um, which are blood vessels. Those of you in physiology already kind of know uh, a good deal of what I'm going to talk about today, um, but we're going to go in a little bit more depth on certain topics. So the the lecture has a number of learning objectives which are listed here, but I've um, differentiated the list of learning objectives based on which part of the lecture. The lecture has it basically has 4 parts. So first, we're going to talk about very general aspects of the structure and function of blood vessels. Then we are going to focus specifically on smooth muscle. We're going to talk about the cell biology of smooth muscle. And then we're going to go straight into the endothelium, which is the other main cell that's found in blood vessels. They're not the only two, but smooth muscle and endothelia are sort of the key players here. So in the third part of the lecture, we're going to discuss endothelial cells. And we're going to wrap it up with 2 important functions of blood vessels that are nested in the endothelium. So it's sort of like endothelium part 2. But we're going to talk about angiogenesis and the regulation of barrier function. So for the first part, which is basically the general principles of structure and function, we're going to start with what I like to call the original problem here. So take, for instance, this hypothetical unicellular organism, right? This is a stock AI generated picture, so this does not really represent any single particular organism, but think of something that's a single cell swimming in the ocean, doing what it does, right? This living organism requires the same things that humans do, right? Nutrients, and it produces, as part of its metabolism, it produces waste, right? And if you produce waste, you have to eliminate it, because when waste accumulates, things go wrong, right? So in this particular scenario, the unicellular organism performs this exchange directly with its environment, right? So some of the, if this were an aerobic organism, the oxygen can diffuse directly into the cell, where it can be picked up by structures, say mitochondria or something like that, that use this oxygen, right? The nutrients, the glucose or whatever other nutrient molecule that this organism is using, can directly enter the cell via transporters or via endocytosis. All of the things that cellular biology, all of the tools that cellular biology has, this organism can and usually does utilize, right? But the important thing is that this exchange of nutrients and waste occurs directly with the extracellular environment, right? So now let's move up a few 100 million years in evolution. We can move from unicellular organisms to multicellular organisms that are still microscopic, right? But here we have a few hundred cells, right? In certain cases, if the organism is multicellular but still small enough, the exchange can still occur between the environment and what we call the parenchyma. So this is actually a moss leaf, right? So a plant. But in this scenario, what we have is the generation of a gradient, right? So because in this case, we're going to focus on oxygen. Oxygen is, let's say, highly concentrated outside of the organism, and then it readily diffuses to the outer layer of cells. But the deeper you go, the less oxygen can diffuse, because, as you probably know, diffusion is much less efficient at longer distances, right? So the molecules of oxygen cannot get to the deep section of the organism as easily as they can reach the outer side, right? So this is actually something that is effectively true in many different organisms, right? But humans are not like any of these two scenarios, right? In humans, and any other like larger organism, particularly multicellular organisms such as animals, right? And to a certain extent, plants as well. We have an issue, right? We have cells that need the nutrients, but they're not even close to being close, so to speak, to the sources of these nutrients, right? So how do we deliver oxygen to the cells that are in the myocardium, for instance, right? So in cells that are in the really deep places in the heart. How do we deliver oxygen to the cells in the liver, right? And by the same token, those cells are producing metabolic waste. How do we get rid of them, right? So the questions that we need to pose are these, like, how do we acquire the nutrients and the necessary metabolic substrates? How do we deliver them to those cells that are far, far, far away from their ability to capture them from the environment, right? And how do we deal with waste elimination, right? And the answer of evolution in this, say, 1 billion years ago or so, maybe less, let's say 600 million years, is the development of specialized structures that can account for the capture, the distribution, the delivery, and the elimination of nutrients and waste, right? In animals, those structures have taken the shape of the heart and the blood vessels, right? And very close to the heart and the blood vessels, we have other structures that are important in the capture and the elimination. So for instance, we can capture nutrients using digestive systems, right? And the digestive system is basically just a tube of structures that enables the process, the capture, the processing, and the digestion of macronutrients, so like macromolecules, into individual monomers, right? And then there is a process of exchange between the digestive system and the circulation, right? Oxygen, right? The gases that we need to the gas that we need in order to run our metabolism is picked up by the respiratory system, right? There are pressure gradients and there's air that moves into the lungs, and then in the lungs there are specific capillaries that enable the exchange of these materials between the alveoli in the lungs and the circulation, right? This is a process of exchange that we can also use to get rid of certain waste, such as carbon dioxide. More waste is exchanged here in the kidneys, right? So the kidneys are also an important structure that deals with waste elimination primarily. But then the things that are picked up by the circulation from the digestive system, from the respiratory system, are circulated through the entire body, and they are delivered. They can be exchanged with structures in the nervous system, structures in the endocrine system, in the musculoskeletal system, so on and so forth, right? So this is to illustrate that the main function of the circulatory system is to establish the system of exchange and delivery, right? Now, specifically, blood vessels enable this transport, but as part of their physiological functions, they are also able to control blood pressure. And those of you in physiology will probably remember that blood pressure is important because it provides the driving force for the movement of the transporting of the conveyor belt in a way, which is the blood. But blood vessels also regulate inflammation. They regulate immune responses, and they have they have organ-specific functions, such as the regulation of metabolism in adipose tissue and skeletal muscle. And there are specific organs in the body that establish particularly strong barriers between the blood and the tissues, right? Those barriers are important in the brain. It's super important that the brain doesn't receive, that the brain doesn't basically accept things that it doesn't need, right? So there are very few things that can cross the blood-brain barrier. And in the testes, for instance, there's a really important barrier there because, and the testes and the ovaries, actually, I should update this with the ovaries, because that's where gametes are produced, right? So egg cells and sperm cells. And if you remember, egg cells and sperm cells have undergone a special type of cell division called meiosis. And that means that the product of that special cellular division are cells that are genetically distinct from the parent organism, right? And if they're genetically distinct, they express different proteins. And if they express different proteins, then they have different antigens that can be potentially recognized by the immune system, right? So the blood testes barrier, for instance, is important in preventing that the immune system attacks sperm cells that technically don't belong to the organism, right? So, and there's a philosophical question there for you, but that's all I'm going to say about that. Okay, so in terms of structures, there are mainly 5 types of blood vessels. We classify them in arteries, arterioles, capillaries, venules, and veins. And this distinction is based on the structures of these particular blood vessels. So this grid basically shows you the general the range of diameters of these blood vessels, the mean thickness of the wall of these blood vessels, and these graphs on the right side basically tell you the relative proportion of the different tissues that are found on blood vessels. So there are basically 4 types of tissue, from deeper to superficial, so that's from inside towards the outside. All of them have an innermost layer of endothelium, which is indicated by that innermost layer of purple cells in the cartoon. Only some of them, so arteries, the larger arteries and the larger veins, contain a decent amount of elastic tissue, so they have the ability to stretch and recoil. Arteries, arterioles, and veins contain smooth muscle, and it's important to understand that arteries and arterioles have smooth muscle in higher proportion than veins do. And fibrous tissue, basically this is a type of connective tissue, is present importantly in arteries, venules, and veins, right? And you can tell that, for instance, a couple of interesting things. Arterioles basically are made of endothelium and smooth muscle, very little fibrous tissue, no elastic tissue. Venules are mostly made of endothelium and fibrous tissue, and capillaries are only made of endothelium. So it's a single layer of endothelial cells. And we're gonna talk about them a little later. Now, a few features of each of these groups of blood vessels. Arteries basically have the ability to vasoconstrict in response to stimulation with alpha-1 adrenergic agonists, right? So anything that binds to alpha-1 adrenergic receptors will cause smooth muscle vasoconstriction, right? Smooth muscle contraction in these arteries. And the same thing is true for arterioles, by the way. Arteries and arterioles can also exhibit a degree of vasodilation. So in other words, relaxation of that smooth muscle that's mediated by a mediator. It's mediated by this molecule that we're going to talk about later called nitric oxide. Arterioles functionally exhibit both of these, the same functional features as arteries, but they are important because they deliver blood to capillaries, right? So blood is transported in these large arteries. These arteries basically start branching out into smaller and smaller arteries. Those smaller arteries then branch out further into arterioles, and the arterioles ultimately deliver blood to the capillaries. That's not what I wanted to do. For some reason, we are not sharing the screen anymore. There we go. So now capillaries have very thin walls. As you can tell, you know, the thickness of the capillary is basically half a micrometer, and actually that's the thickest part of the capillary wall. There are parts in which the capillary wall can be 0.2 micrometers. And they're very tiny. So you see that the diameter of these vessels is between 4 and 10 micrometers. Just so you have an idea, the average diameter of a red blood cell is somewhere between 7 and 8 micrometers. So in the smallest capillaries, the red blood cells have to sort of squeeze in, right? And this is important because this thin wall is ultimately what enables diffusional exchange of gases and waste and nutrients between the blood and the tissues. Venules are primarily tasked with collecting blood from the capillaries, and then they these venules basically merge together and form larger venules that then merge again and form ultimately the larger veins. And veins are basically tasked with carrying blood back to the heart because of a number of reasons that we don't really have time to talk about today. Venules and veins are low pressure vessels. So in order to move blood through this low pressure system, you need to have valves that prevent backflow, right? So if the pressure is not too high, it's hard to bring blood, for instance, from the toes back to the heart because all of that movement needs to happen against gravity, right? Gravity is sort of pushing blood down towards our toes, and the presence of these valves prevents blood from just pooling in our toes when we're standing up. Now, this is a sort of a figure that shows how are these things organized together, right? So you have arteries with all four tissue layers, right? So you have endothelium, you have the smooth muscle layer, you have the adventitia, which is the fibrous tissue, and then in this particular case, there is the elastic lamina. The larger veins basically have the same structure with a couple of differences, but generally speaking, the organization of these tissues is the same. Arteries branch into larger arteries, and then these branch into arterioles. The arterioles give rise to capillary beds, and you see that the capillaries are very well interconnected. In the capillaries, if you take the cross-section of a single capillary, you see that the capillary is made of a single layer of endothelium with a little bit of fibrous tissue. That's it's a different type of fibrous tissue, so ignore it for now. And with respect to the difference in size, you see that the red blood cell occupies most of the size, most of the diameter of the lumen of that tiny capillary. Capillaries then drain into venules. Venules merge together to form larger veins, and then the veins basically bring blood back to the heart, right? And we are going to talk a little bit about how the capillaries mediate the exchange of materials a little later in the lecture. Now, as I said before, the vascular wall is primarily made of 3 layers. So from a histological perspective, we identify three layers. 3 layers is not the same as, so these 3 layers are sort of anatomical histological terms. The 3 layers contain the 4 types of tissue. That's ultimately the idea that I wanted to convey. So the tunica externa, or the adventitia, is basically formed by the fibrous tissue layer that's in the outside of most blood vessels, right? And particularly in arteries and veins. Tunica media contains the smooth muscle smooth muscle cells, right? And the tunica intima contains the endothelium, and it contains the elastic tissue in the vessels that have elastic tissue, right? So that accounts for the 4 tissue types, right? We have 4 tissue types: fibrous, smooth muscle, elastic tissue, and endothelium. The tunica intima contains 2 of them, right? The endothelium and the internal elastic lamina. All right. And this is sort of how they are organized. You see that in particular, I want you to pay attention to the way that the smooth muscle cells are organized, because in blood vessels, the smooth muscle is organized around, right? Around the circumference or around the lumen, so that this means that when those smooth muscle contract, the lumen diameter changes, right? So if the smooth muscle layer exhibits contraction, the lumen diminishes. The lumen becomes smaller. If the smooth muscle dilates, right, or relaxes, then the lumen increases in size, right? And that's a really, really important feature of the arterial circulation in particular. Cool. So we're going to go straight into part 2 because we can. And since we were talking about smooth muscle, this is actually a good segue into it. So there are 3 types of muscle in the animal body, right? At least in the mammalian body, and in many others. But and those are skeletal muscle, cardiac muscle, and smooth muscle, right? You probably have heard a lot about skeletal muscle. When we teach about skeletal muscle, this is where we talk about sarcomeres and blah, blah, blah. Did you have the heart lecture already with Dr. Babu? Yeah. So he probably talked a lot about cardiac muscle. And you probably understand that cardiac muscle is, whatchamacallit? Cardiac muscle is basically a smaller version of skeletal. So there are a few other features, but they contract in the same way, right? Now, smooth muscle is very important because it shares with cardiac muscle the property of being involuntary, right? So you cannot control voluntarily, you cannot decide to control smooth muscle. You can decide to control skeletal, but not cardiac or smooth muscle. But the contractile machinery in these cells is different, right? The proteins are basically the same, but they're not organized in the same way. In blood vessels, the function of smooth muscle is to control vessel diameter, right? So at the center, you have a normal or resting blood vessel. If there's a signal for the smooth muscle to contract, the vessel diameter becomes smaller. If there's a signal to vasodilate, the vessel diameter increases, right? And these changes are really, 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 really important in the regulation of blood pressure, the regulation of blood flow, and the control of resistance, basically. Under the microscope, the smooth muscle looks a little bit like this. So you don't see any striations. The cells are uninucleated, and they're very packed together, and they form like, they have the what we call spindle-like shaped, right? So they have this shape of like a spindle. In culture conditions, so if you take them out of the body and you put them in a petri dish, technically not a petri dish, but tissue culture dish, they basically look like this, right? So they retain some of that spindle-like shape in culture conditions. Now, if you were to do an ultrastructural analysis on these cells, what you would find is that the sarcoplasmic reticulum, so this sort of modified endoplasmic reticulum of these cells, actually localizes to the periphery of the cell, right? So it's relatively close to the membrane. And in particular, it tends to congregate in areas of the cell that exhibit these invaginations called caveolae, right? So these sort of like dips in the cell membrane that you see there, those are called caveolae, right? And caveolae are features in many different cell types. So smooth muscle cell has a lot of caveolae. We're going to talk about caveolae also in the context of endothelial cells. So again, the idea is that these caveolae basically serve as organizing sites for the sarcoplasmic reticulum in these cells. Any questions so far, by the way? No? All right. So the contractile function of smooth muscle cells depends on the organization of contractile proteins. And the contractile proteins are basically the same type. They're not exactly the same proteins that we've seen in skeletal or cardiac, but they act basically the same. So there's actin and there's myosin. The actin in smooth muscle and the myosin in smooth muscle are slightly different than those found in other cell types, but they are still actin and myosin, right? So they're slightly different versions of them. And as you can tell, the proteins are not organized in sarcomeres, right? So you don't have Z discs, you don't have A bands and H zones and all of that stuff. However, you do have these sort of bluish purple blobs. They represent structures that are called dense bodies. And dense bodies contain several scaffolding proteins that are kind of akin to the structural beams in a building, right? So these are basically thick anchoring pillars that the actin filaments can actually attach to, right? And then the myosin filaments are sort of found in between, so that when myosin contracts and when myosin generates a power stroke, the myosin filaments, the myosin heads basically pull on actin very much in the same way that they do as they do in other cell types. The difference is that since you don't have those Z discs, and therefore you don't have sarcomeres to contract, what happens is that the cell basically contracts kind of in every direction, right? So it's like you're having your, think of my arms here, and probably in the recording you can see them, or from home you can see them. My arms represent actin filaments. My torso would represent sort of a dense body, I guess. And then the action of the myosin molecule basically brings all of this together, right? And because some of those actin molecules are kind of connected to the membrane of the cell, the contraction of the smooth muscle cell basically looks like the cell has shriveled. If you take a single smooth muscle cell, the cell basically looks like it has shriveled and shrunk, right, in all possible directions. Of course, that's what happens to a single cell, but these cells are connected to each other with junctions and all sorts of things. So the tension gets distributed all around the tissue. And when that happens in the context of a blood vessel, then this contraction that you observe here turns into the reduction in lumen size, right? So what are the pathways that contract? This is, after all, a course in cell biology, so signaling pathways are at the order of the day. So there are basically two ways to contract smooth muscle, right? And in particular, vascular smooth muscle. The first pathway depends on calcium, right? And if you remember, if those of you who were in the smooth muscle lecture in physio, will remember that we talked about pharmacomechanical coupling and electromechanical coupling. Both pharmaco and electromechanical coupling depend on calcium, right? So whatever the original trigger is, so whether it's electrical stimulation and in the form of an action potential, or an agonist binding to a receptor, both things turn into an increase in intracellular calcium, right? Now, this increase in intracellular calcium can be, as I mentioned before, elicited by binding of a GQ-coupled receptor, right? And this is the case of norepinephrine that binds to alpha-1 receptors. Angiotensin 2 does the same. Anything that leads to the depolarization of the cell and thus triggers the opening of L-type calcium channels. There are in some situations, stretch will lead to the opening of mechanosensitive channels, such as PSO1 and TRPV4. You don't need to remember the specific detail of each, but those are names and molecules that keep coming up for various reasons. And in certain cases, pro-inflammatory agents can also activate GQ receptors, GQ couple receptors. So these are the general 4 ways to increase intracellular calcium or cytosolic calcium in these cells. When any of these things happen, the increase in cytosolic calcium leads to the formation of the calcium calmodulin complex, right? And then the activated calmodulin is able to activate a kinase called MLCK, myosin light chain kinase. As the name kind of suggests, myosin light chain kinase phosphorylates the light chain of myosin. And the light chain in myosin molecules is a regulatory site of the molecule that basically regulates its ability to hydrolyze ATP, the rate at which the molecule is able to process ATP. So when myosin is phosphorylated, this rate of activity basically gets increased, and it's more likely that in the presence of ATP, the myosin molecule will basically generate more power strokes per unit time, and overall, that's going to increase the tension generated by the muscle, right? So again, remember, anything that leads to an increase in cytosolic calcium in smooth muscle cells will activate calmodulin. Calmodulin stimulates MLCK. MLCK phosphorylates myosin, and that increases tension. All right? There is another slightly more obscure way to contract smooth muscle, and that is the Rho kinase pathway. ROC stands for Rho activated kinase. Don't ask me who gave the name, but whatever. Just assume that it's a kinase that is activated by Rho. Rho is a type of small GTPase that acts as a molecular switch, right? So basically, these small GTPases basically switch between the GDP bound form, which is usually inactive, and the GTP bound form, which is usually active. And the transition between the GDP and GTP states is mediated by either GEF, so guanine exchange factors, that basically stimulate the exchange of GDP for GTP, and that process turns the protein on. And then GAPs, or GTPase activating proteins, basically stimulate the GTPase to hydrolyze GTP and release the phosphate, which basically turns it off because as a result of that, the protein becomes GDP bound. So one of the effectors of activated Rho is, in fact, Rho kinase, right? So Rho GTP is able to activate this kinase, which then leads to the phosphorylation of a bunch of things. And this is basically what I was just telling you, right? So the switching between GDP and GTP is regulated by these enzymes. And in the specific case of Rho, there's a particular G couple protein, a G protein couple receptor. One of the obscure ones that we didn't really emphasize I mentioned it, but we didn't really emphasize it in the signaling class. G1213. G1213 coupled receptors lead to the activation of a GEF called P115, right? And the activation of P115 basically turns inactive Rho into active Rho. And the active Rho is able to bind the kinase, this Rho kinase, which is represented by this. The activation of the Rho kinase then leads to the phosphorylation of, its substrates, and those substrates are many different proteins. So how does all of this lead to contraction of smooth muscle? So imagine that this receptor is actually a G1213 couple receptor. That's going to lead to the activation of P115 GEF, right? And that's going to activate Rho A in this particular case. There are many, many different types of Rho, but Rho A is the one that we care about now. So activation of Rho A leads to the activation of Rho kinase, ROCK, right? And activated Rho kinase has two sort of synergistic functions that lead to contraction. Both of them involve the they involve the inactivation, and this, hear the words because this is important. The active Rho kinase leads to the inactivation of a protein called MLCP, not MLCK, MLCP, which stands for myosin light chain phosphatase, right? Myosin light chain phosphatase basically does the exact opposite to the activity of the kinase, right? So kinases and phosphatases are antagonistic to each other. Kinases phosphorylate, phosphatases dephosphorylate. They remove phosphates from molecules, right? So if you think MLCK phosphorylates myosin and increases its activity, MLCP dephosphorylates myosin and it reduces its activity, right? So the state of contraction in smooth muscle, generally speaking, it's sort of hanging in the balance of how much MLCK activation you have and how much MLCP activity you have. If there is more MLCK activity, that leads to contraction. If there is more MLCP activity, you have less phosphorylation, and that leads to relaxation of the smooth muscle. Does that make sense? Again, when you have remember that the contraction of actin and myosin depends on the phosphorylation of myosin, right? If you phosphorylate myosin, that leads to contraction. If you remove the phosphorylation, that leads to relaxation. Phosphorylation of myosin depends on MLCK. Dephosphorylation depends on MLCP, right? So the balance between the activity of these two is ultimately what determines the level of phosphorylation of myosin and ultimately the ability of myosin to generate the power stroke and lead to contraction. So what Rho kinase does is it promotes the dephosphorylation of MLCP of myosin light chain by basically inactivating myosin light chain phosphatase. So I just contradicted myself. I'm going to start over again. Rho kinase inhibits MLCP. If you inhibit MLCP, you have less dephosphorylation and more phosphorylation, and that leads to contraction. Does that make sense? Sorry for the mix-up. It's been a long day. Any questions about this? This is a little complicated. This is allegedly one of the complicated things in this class. Yes. The question is whether this pathway controls how long the contraction lasts for, and that really depends. This is more of an acute phenomenon, but the continued activation of these receptors, for instance, can lead to a certain tonic state of contraction, right? So if overall, if the cell can, for instance, afford to produce all of the ATP that is necessary to keep calcium high and to keep myosin turning over, then yes, right? But that's not necessarily something that all smooth muscle cells want to do all the time. Does that make sense? Any other questions? No? Okay, so this graph is a little weird, but basically, this paper, which is from 1997, so you see almost 30 years ago, this paper basically showed that inhibiting Rho kinase leads to vessel relaxation. So they created this group basically was trying to characterize the functions of a particular molecule which they call Y27632. This molecule is a very well-known inhibitor of Rho kinase, right? So that's what it does. It prevents Rho kinase from doing its job. So in this graph, what you have is percentage relaxation. So this is a normalized measure of relaxation as a function of inhibitor concentration, right? And the concentration is expressed in a logarithmic scale. So what they did was they took the aorta from rats, right? They cut the aorta from several rats in tiny little rings, and they put these rings into a little bath and measured the ability of the rings to contract or relax, right? So they did this in the presence of various concentrations of the inhibitor, but in order to remember that what they were measuring was relaxation, right? So in order to measure relaxation, you had to pre-contract the muscle, right? And they did this in two ways. They pre-contracted the muscle by bathing it in a solution of potassium chloride, right? So basically this solution of potassium, this is a really, really high concentration of potassium chloride, causes the full depolarization of the cell, opening of these voltage-gated calcium channels, and therefore these cells are basically full of calcium all the time, right? The other thing that they did is to apply an alpha receptor agonist, which is called phenylephrine. So in the presence of phenylephrine, the phenylephrine basically activates the alpha-1 receptors, and that triggers contraction, right? So what they did was to, in these pre-constricted rings, they applied the different concentrations of the inhibitor, and you see that in both cases, the higher the concentration of the inhibitor, the higher the percentage of relaxation, right? And this means the more you inhibit the kinase, right? The more you inhibit the kinase, the less the cells can contract, right? The more you inhibit the kinase, the more they can relax, in other words, right? You see that I don't understand why they decided to invert the y-axis, because it's sort of counterintuitive. But you see that 0 is at the top, and at 10 to the negative 5 molar of inhibitor, you have almost maximal relaxation in the phenylephrine constricted rings, right? The effect is not so good in the potassium chloride constricted rings. Why would that be the case? Why would that be the case? It has to do with calcium, yes. But more so than with calcium by itself, it has to do with the source of calcium, right? Because in the phenylephrine constricted rings, basically what phenylephrine is doing is activating this side of the pathway, right? So the calcium that contracts the cell is coming from the sarcoplasmic reticulum. And inhibiting Rho kinase basically counteracts all of that. But if you depolarize the cell, only part of that calcium is coming from the sarcoplasmic reticulum. a significant amount of the calcium that is contracting these cells is coming from the extracellular fluid. And that the Rho kinase can't quite overcome. It helps, but it amounts to only, 40% of the total contraction, right? So that's how you should take a look at this, this particular figure. Any questions? All right. So as I was saying before, the contraction of smooth muscle depends on the dephosphorylation of myosin light chain. So the idea is that in order for smooth muscle to relax, two things need to happen. You have to first have a decrease in cytosolic calcium concentration. The amount of calcium in the cytosol of the smooth muscle cell needs to come down. The second thing that you need is to dephosphorylate the light chain of myosin, right? So the first part, so the decrease in intracellular calcium depends on transporters. The sarcopump in the sarcoplasmic reticulum that is pumping calcium back into the sarcoplasmic reticulum. And depending on the tissue type, the plasma membrane calcium ATPase that pumps calcium to the extracellular fluid, and the sodium calcium exchanger, right? These two things basically do the same thing. Number two depends on this balance that I was telling you about, right? The balance between the activity of MLCK and MLCP. So to dephosphorylate myosin, you have to either decrease the activity of MLCK or increase the activity of MLCP. Both of these things will lead to a decrease in myosin light chain phosphorylation. And importantly, in resistance vessels, so that means the smallest of arteries and the majority of arterioles, the changes in MLCP and MLCK activity are almost directly the result of nitric oxide production in the endothelium. All right? In the endothelium. And we're going to talk about this mechanism a little later. Now, the smooth muscle cells are important in vascular biology and in cardiovascular biology generally because they mediate two important processes that are associated with disease states, right? So generally speaking, in the adult healthy individual, smooth muscle cells are found in what we call a quiescent state. These cells are fully differentiated, they're fully mature, and they don't really need to enter or exit the cell cycle, right? They're happy doing what they do. Under some circumstances, though, some of these cells can actually de-differentiate into something that we call a microproliferative state, right? And the balance between these two states depends on the extracellular matrix, growth factors, and the ability of the cell to establish good connections with their neighbors, right? Via adhesion receptors. And that adhesion basically speaks to the ability of the cell to attach to its neighboring cells, but also to the extracellular matrix. So in pathological situations such as atherosclerosis or arteriosclerosis, those two things are different. What usually happens is there is a movement from the quiescent to the microproliferative state, right? And this phenotypic switching basically depends on changes to the extracellular matrix, the presence of growth factors, and changes in the activity of these receptors, right? Atherosclerosis, which is this pathological a pathophysiological process in which there's an infiltration of inflammatory cells, and it's sort of an inflammation of the vascular wall that leads to the formation of this atherosclerotic plaque that tends to be infiltrated with lipids and, again, cells of the immune system. And it's associated with smooth muscle cells basically acquiring this migration feature because they move around, right? Arteriosclerosis is a different story because this is something that's called neointima formation, right? In other words, the smooth muscle cells acquire a proliferative phenotype and they begin to basically multiply. But they don't multiply outwardly. They actually multiply inwardly. And that leads to a drastic reduction of the lumen of that blood vessel. And that process is called neointimal formation. So happens that if you give Rho kinase inhibitors to rats that have suffered injury in their blood vessels, inhibition of Rho kinase actually prevents the progression of this pathological state. So what the authors of this paper did was to basically they blew up a little balloon inside the carotid artery of these rats. So basically what happens is that they caused stretch of the carotid arteries beyond the ability, beyond the physical capability of those arteries, and that caused injury, right? So how do you repair injury in a blood vessel? You have to, some of these cells actually have to enter this proliferative state to basically patch the damage, right? So this cell here, so in this particular rat, this is no injury, so this is what we call a control. And then they caused injury and treated the rats with what we call vehicles. So that's, again, this is another control. So you see that the injury caused the thickening of the smooth muscle layer, right? The smooth muscle layer is seen in this sort of like dark brown layer. And when you give hydralazine, which is basically a vasodilator, you see that a vasodilator molecule cannot cause vasodilation in these cells. So in other words, these blood vessels are compromised in their ability to do what they are supposed to do. However, when they caused injury in rats that were treated with Rho kinase inhibitors, and that's panels D and E, you see that there's no significant proliferation, right? So what this data suggests is that inhibiting Rho kinase prevents, in this case, neointimal formation. It, in this case, would prevent arteriosclerosis. Does that make sense? And this panel here only represents, they did Western blots to basically look at the activity of Rho kinase targets. So you see that the darker the band, that means that the higher the activity of these things. And when you block Rho kinase, then you don't really have that activity. And so this is sort of a confirmation that these things are happening. But that's basically it in terms of smooth muscle. Any questions? Nope. All right. So let's take a 10-minute break. Let's resume at 7:07. Sounds good? All right. So let's continue. Okay, so part 3, we the first hour we basically spent talking about parts 1 and 2. Part 3 is about endothelial cells. So I actually need to click here. All right. So endothelial cells form a monolayer that lines the lumen of all blood vessels and the chambers of the heart, right? So it's a continuous monolayer of endothelial cells that lines the inside of every blood vessel and all of the heart's chambers, right? In the heart, the endothelium is called the endocardium. So just so you know. When you put all of these endothelial cells together, basically all of this amounts to a really, really, really large organ, right? So you have about 1 kilogram, so a little over 2 pounds worth of endothelium in your body. So similar to the size of the liver, and the liver is one of the bigger organs in our abdominal cavity. And if you put all of these endothelial cells together and you line them up on the floor, you'll get a surface area that's equivalent to 8 tennis courts. So this is a massive amount of cells. Now, the vascular wall is the organ that we're mostly concerned with today, and it includes the smooth muscle, but it also includes the endothelium. So in the larger vessels, the endothelium forms this layer that's called the tunica intima, and the other layers are basically the adventitia, the elastic fibers, the smooth muscle, and all that stuff. Now, something that's very particular about the endothelium is that it's called it exhibits this property that is called organotypical cell. Or, in other words, the function and the property, sometimes even the structure of these endothelial cells depends on the organ where they are found. Because let's put it this way, the needs of transport, so the needs, or let's put it this way, the barrier that is established between the blood and the brain, right, doesn't necessarily have to be the same as the type of barrier established between the blood and the liver or the blood and the skin, right? So because the endothelium forms that barrier and it actually regulates that barrier function, the endothelium adapts to the organ where it is in order to fulfill its role, right? So when you take a look at a blood vessel, and this basically represents either a very small venule or a capillary, the endothelium looks like this. So these endothelial cells form junctions between each other. They wrap themselves around the lumen of the blood vessel. And in the case of capillaries, there's usually 1 or 2 endothelial cells worth of forming that lumen, right? In larger arteries, of course, you have more. You have many more, right? But in the tiniest of blood vessels, the lumen is formed by 1 or 2 endothelial cells. And they organize by forming this sort of cobblestone pattern, right? So in this exterior view, this is what you would see if you took a look at the if you looked at the capillary from the outside. And if you sectioned the capillary, you would see that the inside of the capillary looks very similar, with the nuclei in these endothelial cells being basically the sort of the thicker part of the cell. This picture was captured by my PhD mentor in this very building, actually. back in 1979. So this is an electron micrograph of a post-capillary venule in the heart of a dog, right? Back then, it was very common to experiment on dogs. Today, not so much. But basically what you have is the post-capillary venule around here, and this darker blob at the center is a red blood cell that is sort of like twisting itself in order to fit through that very narrow post-capillary venule. So this line here basically represents the outline of the post-capillary venule, right? If I remember correctly, there are 2 or 3 endothelial cells forming this post-capillary venule. 3, in fact, labeled there, ECs. Many capillaries and blood vessels in general, but particularly capillaries and venules, exhibit these satellite cells called pericytes that basically function to support metabolically, and they maintain the state of differentiation of the endothelial cells. And because this is the heart, what you have on the outside is cardiac muscle. And you see that all of those tiny little dots are basically thin and thick filaments. So those are myofibrils in the contractile myofibrils in the cardiac muscle. You can see a couple of mitochondria there and there. And these blue arrows depict the junctions between these endothelial cells. And you see there's a junction there. There's a junction there that has a weird shape, and there's another junction there. So that's how I could tell that there are 3 endothelial cells in this section. I told you about caveolae earlier, right? So if you pay close attention, you will see that there are many of these tiny little invaginations, and I marked a few of them with those brown arrows, facing the outside of the endothelial cell. In pink, the arrows, so the pink arrows indicate what we call luminal caveolae, right? So these are caveolae that are actually facing the lumen of the post-capillary venule. So this basically, and you know, if you are able to see, I actually don't know how much you're able to see. They're visible, I think. You folks at home probably see them better. It's a little better. The reason why I'm pointing out the caveolae is because they're a really, really important structure in the endothelial cell. The caveolae, whether it's in smooth muscle or endothelial cells, they're always basically these invaginations of the plasma membrane that have a lot of this protein called caveolin, which is basically the protein that sort of gives the caveola its sort of shape. And they're particularly interesting because these are regions of the membrane that are enriched in particular forms of lipids, particularly sphingolipids and cholesterol. So if you may have heard about in other courses, or maybe in this course too, you may have heard about lipid rafts before. Lipid rafts are these regions of the plasma membrane that have especially high concentrations of some lipids, including cholesterol. So these caveolae tend to locate in areas of the membrane with a lot of lipid rafts. The important aspect of this is that these caveolin in particular serves as a scaffold for several receptors and intracellular signaling molecules. So these are sort of signaling hubs in the cell, and therefore they are important in controlling several functional aspects of the cell at the molecular level. Now, why do we talk about the endothelium so much? Turns out that the importance of the endothelium in controlling for vascular function was sort of an accident, or at least it was discovered by accident. So in 1980, Robert Furchtgott and John Sawatzky, working in Stony Brook, so not too far from here, they basically published this paper in Nature that described the obligatory role of endothelial cells in the relaxation of arterial smooth muscle by acetylcholine. So what, at this point in time, it was known that acetylcholine, which is a molecule that's released by parasympathetic neurons, but also by a bunch of other cells. Acetylcholine caused vasodilation. That was a known fact. If you put acetylcholine in a blood vessel, an artery or an arterial, that blood vessel was going to dilate, right? So what was known was that acetylcholine caused relaxation of smooth muscle. And by accident, actually, they discovered that in their preparations of the aortic rings from rabbits, they were doing a bunch of different experiments to test for something completely different. And they were using this property of acetylcholine to vasodilate. And for some reason, they discovered that the acetylcholine that they were using was not working, right? And they troubleshoot the way that scientists like to do troubleshooting, which is an elegant way to say that we hate doing troubleshooting because it's terribly boring. But you're trying to get things done, and you need to work out why the experiment is not working the way it should. So basically, they discovered that in the preparation of these aortic rings, whoever was doing the experiment was actually rubbing the aorta so hard that the endothelium was actually destroyed, right? So effectively, their preparation did not have an endothelium. So when they discovered this, they actually started testing this. And they figured out that in this particular experiment, the black curve represents tension, right? So the higher the tension basically means the higher the level of contraction, right? So they applied noradrenaline, right, norepinephrine, and the application of norepinephrine increased tension, so it caused contraction as it should. And then they applied acetylcholine at increasing concentrations, so 10 to the -8, 10 to the -7, 10 to the -6, so increasing concentrations of acetylcholine. And the more acetylcholine you put, the more it dilated, right? So this is your control. Now, when you rub the aortic ring and you basically destroy the endothelium, and then apply norepinephrine, you contract, but then if you keep putting acetylcholine, is there any relaxation? No, right? So basically they published this and they said the endothelium is necessary for the relaxation of blood vessels mediated by acetylcholine, right? And they did all of this in the late 70s, and they published their paper in 1980. They called this endothelium factor, they called it EDRF, endothelium-derived relaxing factor. And at this point, they had no way of knowing what the chemical nature of this factor was. A lot of work followed this discovery. And in June of 1987, Palmer, Ferrige, and Moncada published this paper in which they identified the endothelium-derived relaxing factor as nitric oxide, right? And I'm not going to show you the details of the papers because it's boring and sort of arcane, but they published this. In December of the same year, 1987, in a different journal, Louis Ignarro and his colleagues published another paper in which basically they claimed the same thing. Endothelium-derived relaxing factor produced and released from artery and vein is nitric oxide. So In other words, a completely separate group of investigators discovered the same thing, which is something that happens quite a lot in science, right? People discover things independently of each other. Q to 1998, and these people won the Nobel Prize for these discoveries, right? The prize was given to Furchtgott, Ignarro, so I showed you papers published by these two, and then by Firat Murad, who basically worked on elucidating what was after nitric oxide in the pathway. So this guy basically discovered cyclic GMP. Who's missing here? Moncada, the Hispanic guy. I mentioned this because there's been a lot of in the history of vascular biology, this has been sort of a sore point for many people because Moncada and Ignarro basically discovered the same thing sort of at the same time. And in fact, Moncada published it before Ignarro. And like I said, there's been a lot of controversy because Moncada, in the eyes of many people, myself included, Moncada should have been also a recipient of the Nobel Prize. But the Nobel Committee basically can only really, within the rules of the Nobel Prize, you can only award it to 3 people. And they have to be alive and blah, blah, blah. So ultimately, the Nobel Committee decided that Moncada was meritorious, that his contribution was worth receiving the prize, but they couldn't do anything else. So they basically snubbed him. So this is a very fascinating story that I like to tell in the context of this discovery because even though, Moncada was born in Honduras, but most of his scientific career was actually, it happened a little bit in Honduras, but significantly in the UK. So he had access to do all of this work. He had access to the resources in a what we call first world country, whatever. But again, it's from as a Hispanic person myself, I would like to highlight these parts of history. And this is not to mention the multitude of people who worked with them that were people of color, right? So there's been people of color in science since the beginning, right? Since the beginning of modern science. And it's important to highlight what those contributions are. Anyway, I digress. We're going to talk about nitric oxide now. Nitric oxide is the central molecule in vascular homeostasis. It's a gas, right? And as a gas, it's also a free radical gas, right? Which means it's very labile. In other words, it's not very stable. That's sort of a definition of labile. Now, what are the functions of nitric oxide? It relaxes vascular smooth muscle, as we just saw demonstrated by Furchtgott and many other people later. And because of this, it causes vasodilation, and thus it helps control blood flow and blood pressure. Nitric oxide produced by the endothelium also prevents platelets from sticking to the wall of blood vessels. And this is important in preventing unnecessary coagulation, right? Nitric oxide basically prevents us from getting blood clots that shouldn't be there. It has an anti-inflammatory function by preventing the adhesion of leukocytes to the endothelium of healthy blood vessels. And it also has a pro-inflammatory function. In sites of injury, it chiefly regulates what we call the permeability of blood vessels to macromolecules, which is an important aspect of every inflammatory response. And we're going to talk about a little, we're going to talk about that in a little. The enzyme that produces nitric oxide in the endothelium is the endothelial nitric oxide synthase, or eNOS, right? There are 3 nitric oxide synthases, each of them encoded by a different gene. Those genes are called NOS1, NOS2, and NOS3. And they encode for, in order, they encode for the neuronal nitric oxide synthase, the inducible nitric oxide synthase, and then NOS3 encodes for the endothelial version. They're not restricted to those types of cells. So the neuronal nitric oxide synthase is present in neurons, but also in cardiomyocytes. The inducible one is, for the most part, only expressed in activated macrophages, so in cells of the immune system. And the endothelial version of it is primarily only in endothelial cells. The endothelial enzyme is a really complex enzyme, and under normal circumstances, it needs to dimerize. So basically, you need 2 molecules of eNOS that bind together to form an active enzyme. The details are not super important unless you want to really study this. But basically, the enzyme contains 2 major domains. It contains a reductase domain and an oxygenase domain. And as an oxidoreductase, basically it catalyzes the transfer of electrons from donors to acceptors. And as a product of that electron transfer, you basically form products, right? Something that's important to mention is that when these monomers, these 2 eNOS molecules, are close but not coupled, the enzyme actually leads to the production of superoxide, which is a free radical that's kind of toxic for the cells. But in the presence of a gazillion different cofactors, including NADPH, a heme group, calmodulin, zinc, tetrahydropterin, and a bunch of other things, what the enzyme does is it takes electrons from reduced NADPH, and it basically puts them into its substrates, arginine and oxygen. So arginine, the amino acid, and oxygen go in, and the products are citrulline, which is another amino acid that's not part of proteins, and nitric oxide. So generally speaking, what I'm trying to say is, if the enzyme is uncoupled, you get superoxide. If the enzyme is properly coupled with all of its cofactors, the enzyme produces nitric oxide, right? And the substrate that is used to produce nitric oxide is L-arginine. Okay? Does that make sense? Any questions? All right. Now, how does nitric oxide do all of that it does? So in this cartoon, basically, the cell on top is the endothelial cell. The cell on the bottom is the vascular smooth muscle cell. So basically, on top here, you have the lumen of the blood vessel. The abluminal side is on this side. So the endothelial cell has many different types of receptors for what we call vasoactive substances. And in particular here, we're going to talk about acetylcholine, which we mentioned already, but also bradykinin and histamine. Those are also vasoactive compounds. Some endothelial cells have receptors for adenosine, for instance, and people in physio will probably remember that we talked about adenosine quite a bit. So activation of most of these receptors leads to the production of, to the activation of phospholipase C and the release of IP3, right? Because these receptors are GQ coupled. So release of IP3 causes release of calcium from the intracellular stores, activation of PKC, and PKC causes further calcium increase by opening calcium channels. Together, both sources of calcium lead to the activation of calmodulin, and calmodulin, again, as I mentioned before, calmodulin is one of the main cofactors for eNOS. So when activated calmodulin binds to eNOS, the enzyme becomes coupled, and then it can basically use arginine and, in the presence of oxygen, turn it into nitric oxide and citrulline, right? Now, this is how chemical mediators cause nitric oxide production. There's another source of, or there's another stimulus for endothelial cells to produce nitric oxide, and that's called shear stress. And shear stress basically is a measure of the force of blood dragging on the surface of the endothelial cell. So think of increasing blood pressure will cause increases in shear stress, because basically the blood is hitting that endothelium harder, right? And that shear stress, through several different pathways, leads to the activation of a protein kinase called AKT, and AKT can also activate eNOS. Ultimately, regardless of what the original stimulus is, nitric oxide is produced, and as you remember, nitric oxide is a gas. And as a gas, it can freely diffuse, right? So nitric oxide is produced, and it diffuses from the endothelial cell into the smooth muscle cell. And in the smooth muscle cell, there is a large amount of a protein called SGC, or soluble guanylyl cyclase, spelled for you there. The soluble guanylyl cyclase is an enzyme that takes GTP and it converts it into cyclic GMP. So this is similar to cyclic AMP, but with a G instead, right? Cyclic GMP is a second messenger that activates a protein kinase called PKG, or cyclic GMP activated protein kinase. And PKG does several things. First, it causes activation of MLCP, right? So if you activate MLCP, what happens to smooth muscle? Relaxation, right? PKG can inhibit MLCK. If you inhibit MLCK, what happens to smooth muscle? Relaxation. PKG will also cause the closure of calcium channels. If you have less calcium entering the smooth muscle cell, you get relaxation. And finally, it triggers the opening of potassium channels. If you open potassium channels, in which direction does that potassium go? Out of the cell, right? And that causes depolarization or hyperpolarization of the cell. It causes the hyperpolarization of the cell, right? And a hyperpolarized smooth muscle cell is less prone to contraction, right? So all of these four outcomes lead to the relaxation of the smooth muscle cell, right? The things that we study the most usually are related to the activation of MLCP, right? So the canonical pathway usually will have you nitric oxide, cyclic GMP, phosphorylation of MLCP, and then dephosphorylation of myosin. The other aspects contribute, but they contribute proportionally less to the overall response. Does that make sense? Any questions about the pathway? Okay, so the enos, I mentioned earlier that enos needs to be coupled in order to produce nitric oxide. And under normal circumstances, that's the standard way that we find enos in. But in pathological states, enos can actually become uncoupled, right? So in the healthy endothelium, the enzyme is coupled. You have the two domains, the oxygenase and the reductase, and you have all the cofactors, including tetrabiopterin, the heme group, the calmodulin, and all of these things that are part of the electron transfer chain. And in the coupled state, you get standard nitric oxide production, which is responsible for vasodilation, platelet aggregation inhibition, reduction of leukocyte trafficking, and then also vascular remodeling for a number of reasons that we're going to talk about later. But if there are situations in which the cofactors are absent, right? So the zinc ion, BH4, calmodulin, any of these, if any of these is absent, the enzyme cannot properly couple, and that leads to the production of superoxide. But the problem is that superoxide is a really, really reactive, it's a really, really reactive oxygen species. And because there is always some nitric oxide around, even in this sort of deceased state, there are some enos molecules are still coupled, not 100% are dysfunctional. When a molecule of superoxide encounters a random molecule of nitric oxide, what you get is the formation of a really toxic molecule called peroxynitrate, which is there. And peroxynitrate really, really causes protein damage. It causes protein nitration, and basically it serves as a sink for nitric oxide. So ultimately, it reduces the overall availability of nitric oxide. This uncoupling of the enzyme ultimately is in the production of superoxide and peroxynitrate causes cellular damage and cell death, right? Now, something that's interesting is that mice that don't have enos, which is something that we can do in the lab, they're prone to exaggerated atherogenesis, so they tend to develop atherosclerosis more frequently, and they have reduced collateral blood flow, which basically means blood has less pathways to flow through. And that makes them vulnerable to ischemia, both in the heart and in the brain. However, they're only slightly hypertensive, which was kind of interesting when this mouse was made back in the mid-1990s. So some work has been done to understand why, if you don't have a major vasodilating molecule, why are you still only slightly hypertensive? Now, the really important thing for most of you, I think, is that diabetes, atherosclerosis, hypertension, and many other diseases, that particularly those that are associated with aging, manifest as impaired nitric oxide production. And this syndrome, generally speaking, is called endothelial dysfunction. So endothelial dysfunction is basically a hallmark of several important pathological states that affect a big chunk of the population. What does endothelial dysfunction do? It's associated with diabetes, associated with hypertension, it's associated with hyperlipidemia, with smoking, aging, obesity, you name it. And in this case, what you basically have is a larger proportion of uncoupled enos, and therefore less nitric oxide production. This makes the body prone to inflammation and to oxidative stress. And in the endothelium, because the endothelium is everywhere, this can lead or at least be a factor in the development of retinopathies. So people can actually develop macular degeneration and retinal hypertension. There can be vascular nephropathy, so kidney damage. Your patients are prone to ACVs, cerebrovascular accidents. For some reason, I gave you the Spanish acronym, which is ACV. Heart problems, angina. So these are like markers of an impending heart attack. The development of heart failure, cardiomyopathy, thrombotic diseases in arteries and veins. And this is where you get pulmonary embolism, for instance, and stroke, and other forms of microvascular dysfunction. So this is just to highlight the fact that the endothelium, and particularly nitric oxide production by eNOS in the endothelium, is really important for cardiovascular and overall homeostasis. Okay? Any questions? Yes. The question is, could this be reversed? There is, because most of this damage that can happen in the vasculature is actually mediated by reactive oxygen species. So in other words, oxidative stress. There's been studies that show benefit of certain supplements such as N-acetylcysteine, because N-acetylcysteine is the most important precursor for glutathione, which is an important reductive agent in the cell. So the rationale for that is, if you load up your cells with glutathione, you are less likely to produce superoxide. And if you don't have superoxide, then you don't have peroxynitrate, and that decreases the damage caused by the uncoupling of eNOS and the low availability of nitric oxide. Now you can loading cells with precursors of nitric oxide per se is a little bit more complicated, because you don't necessarily want nitric oxide all the time, right? But an acute dose of, so for instance, patients that suffer angina pectoris, which is this sort of acute chest pain that is usually associated with a transient period of ischemia in the heart. The first thing that if a patient shows up with chest pain to the ER, the first thing that usually is done is the patient is given a pill of nitroglycerin, right? Which is given to dissolve under the tongue. Nitroglycerin gets absorbed into the bloodstream, and it's enzymatically converted into what we call nitric oxide donors. So not nitric oxide per se, but molecules that produce nitric oxide. And that those nitric oxide donors get to the heart, and they cause vasodilation of the coronary arteries, and that relieves the situation. The problem is that nitric oxide, if you remember, it's a gas. And as a gas, it can diffuse everywhere, right? And a lot of the more recent data on nitric oxide signaling indicates that the timing and the place where nitric oxide is produced determines what the outcome of that nitric oxide production is. So it's not so simple as, I'm going to just give you an injection of nitrate and hope for the best. But you can actually promote the sort of the quenching of oxidative stress with N-acetylcysteine. That's a much more robust strategy. Does that make sense? Yes. Can you say it a little louder? Diabetes. How does diabetes cause oxidative stress? There are several different ways, but one of the things that's particularly important for endothelial. So for you folks at home, the question is, how does diabetes cause oxidative stress? In the endothelium, Diabetes is, again, as you know, characterized by elevated levels of blood sugar, right? And under this type of stress, sort of permanent or chronic hyperglycemia, endothelial cells are more prone to develop something they're called AGE molecules. AGE being an acronym for Advanced Glycation End Products. In other words, many molecules in the cell, particularly those that are exposed to the blood vessel, they acquire chains of sugars that shouldn't be there in the first place, and they become sticky. And when the endothelial cell becomes sticky, a lot of other cells can actually just attach to it. And kind of by chance, that may lead to the activation of several different signaling pathways that ultimately lead to imbalances in the metabolism of the cell. It can lead to the activation of enzymes that actually produce superoxide, there other than enos. And those are the things that I have in mind at the moment. There are plenty of other things that are correlated with diabetes and oxidative stress in the vasculature, but they escape my mind at this point. Cool. Okay, let's take another break before we finish with the last part. So let's resume at 8. Okay. Okay, so I'm going to move a little faster on this part, mostly to avoid digressing on unnecessary details. But there are a couple of important take-home messages from this. So we're going to talk mostly about two complex processes that are mediated by endothelial cells, and those are angiogenesis and endothelial barrier function. So for the most part, the idea is that the endothelium is known as a very dynamic and selectively permeable barrier between the blood and the tissues, right? In other words, not everything that is in the blood needs to move into the tissue, and the structure that regulates who gets to cross over is the endothelium, the endothelial cell. And in the healthy individual, the endothelial cells are similar to the smooth muscle cells. They are quiescent cells. They are highly differentiated. They are contact inhibited. That means that when the cells basically are touching each other, like here, right? They tell each other that, okay, everything is okay. You don't need to keep dividing. So just chill, right? And that makes them non-proliferating, right? They don't divide. And the dynamics of that endothelial barrier at rest, when things are fine and everybody's healthy, is that the permeability of this barrier is low, and the leakage, that means the transport, the flux of matter between the blood and the tissue, is also low. So everything's sort of down. Now, this selective permeability of the endothelial barrier is primarily given by the intercellular junctions, right? That basically create this junction between endothelial cells. But when things do need to cross, and under different scenarios, different things do need to cross. So think, for instance, the transmigration of immune cells, right? So when there is an infection and those circulating white blood cells need to arrive to a site of infection in order to repair the damaged tissue, those cells actually need to cross the endothelial barrier. And in some cases, they do so by basically squeezing in between this intercellular junctions. And in some other cases, they actually poke a hole through the endothelial cytoplasm and they basically move transcellularly. Smaller stuff but still larger than ions, for instance. So we're talking about macromolecules. Quite often leak in between cells via the paracellular permeability pathway, or in some cases, they are basically loaded in the lumen of caveolae that then become vesicles, so they become endocytosed, and they get trafficked from one side of the cell to the other. And in some cases, these, or they're not exactly caveolae, but there's a different type of intracellular vesicle. They sort of pile up together, and they form these, what we call VVOs, or vesicular or vacuovesicular organs, whatever. And under certain circumstances, the membranes between all of these tiny little vesicles basically fuses, and that forms a channel through which things can actually move from one side to the other, right? Those are the general pathways for things to move between the endothelium, between the blood and the tissue. Now, under certain circumstances, certain types of imbalances can trigger an escape from this sort of state of dormancy, right? So this quiescence is lost under certain circumstances. Things that cause a loss of contact inhibition. So for instance, a situation where the intercellular junctions become disassembled, that will basically serve as a signal for the cell to initiate a what we call a transient dedifferentiation. And that tends to activate the cell cycle. So the cell re-enters the cell cycle, and it acquires migratory properties. This is quite often something that occurs in response to prolonged hypoxia, for instance. So a particular section of the tissue that is not receiving the amount of oxygen that it needs, it usually leads to the production of signals that cause all of these things listed here. And the idea is that those signals ultimately cause the endothelium to remodel in such a way that it helps reestablish vascular support to the tissue. And this process is called angiogenesis, right? Angiogenesis basically means the formation of new blood vessels, right? And we're going to talk about how this process happens. When is angiogenesis important? One such example is in the response to critical limb ischemia, right? So if you have a situation of vascular occlusion, so there's something that's blocking blood flow through a particular blood vessel, such that a significant portion of tissue is compromised, right? The lack of oxygen, which is called ischemia, may lead to tissue damage in the form of necrosis. So under normal circumstances, when there's a period of prolonged ischemia, and when blood flow is basically interrupted, the normal response of the tissue is to release molecules that are called proangiogenic factors. These proangiogenic factors basically talk to the endothelial cells, right? And they cause dedifferentiation, they trigger re-entry into the cell cycle, and they trigger this migratory phenotype so that the endothelium can basically remodel itself and form a new blood vessel from a pre-existing one. And as you form a new blood vessel, basically that blood vessel grows in the direction of the tissue that is ischemic. And that basically helps reestablish the supply of oxygen, right? Now, think of a patient that is chronically diabetic, and in which these diabetic complications basically cause endothelial dysfunction, and the endothelium is not able to respond in such a way. So the endothelial dysfunction basically prevents this normal response from being turned on, and you get diminished angiogenesis, you get diminished vasculogenesis, which is a similar but different process. And ultimately, what you get is impaired collateralization of blood vessels. In other words, this means you can't make a new blood vessel when you need one, right? And in that case, you get chronic hypoxia, the tissue becomes necrotic, and then you basically, in order to prevent gangrene and sepsis, you have to amputate the tissue. This is why many chronically diabetic patients end up with amputated organs, right? So the about 10% of the diabetic patients end up with chronic critical limb ischemia, and a significant number of them end up with an amputated limb, which actually is not great for prognosis. Like the 5-year survival rate of patients that have undergone amputation of a limb is only 32%. So again, these are real statistics from real patients, right? So again, endothelium, not because I say so, but the endothelium is kind of cool in that regard, and it's the most important thing ever. Now, the difference between vasculogenesis and angiogenesis. Both processes lead to vessel collateralization. In other words, the formation of new blood vessels. Now, angiogenesis or sprouting angiogenesis amounts for 75% of the formation of new vessels at any given time. And angiogenesis is triggered by the dedifferentiation, migration, and proliferation of pre-existing endothelial cells, right? So these are endothelial cells from an established blood vessel that change their phenotype and lead to the formation of a new blood vessel. Vasculogenesis is more of a developmental process, and it's driven by endothelial progenitor cells that live in the bone marrow and circulate. These are circulating progenitor cells that basically, under the right circumstances, take hold and basically start the process on their own, right? So these are not vessels that originate from pre-existing ones. These are like colonizing endothelial progenitors that trigger the production of new blood vessels. Now, in the particular case of sprouting angiogenesis, the most important thing is the balance between pro-angiogenic factors and anti-angiogenic factors. So when the tissue is healthy, right, normally what you have is endothelial cells producing what we call a basement membrane. So this is sort of the extracellular matrix made by the endothelial cells. And the basement membrane basically attaches to the interstitial extracellular matrix, which is usually made of a lot of collagen. It's made by fibroblasts, by resident fibroblasts in the tissue, so on and so forth. So when the endothelial basement membrane is sort of intact, that basement membrane contains a lot of laminin, right? And the presence of laminin signals the endothelial cell to reduce Rho activity and increase RAC, RAP, EPEC, and pKa activity, right? All of those proteins signal to stabilize the junctions between endothelial cells. They minimize the formation of stress fibers from the cytoskeleton and basically keep the cells in this sort of quiescent state, right? So that's why we call these are the I don't want to use the word ice. Let's go with the word chill. So these are chilling signals to that basically are maintenance signals. They help maintain the integrity of the vessel. Now, when there are pro-angiogenic stimuli, or when there is injury, right? This injury that leads to the disruption of the basement membrane and the exposure of the endothelial cell to the interstitial matrix, right? So you don't have the basement membrane anymore, and the endothelial cell sees this interstitial membrane, which is rich in collagen. Collagen is a signal for an increase in the activity of SARC and Rho, which lead to disassembly of the intercellular junction and the formation of stress fibers in the endothelial cells. When you have stress fibers forming in the endothelial cells, that's a signal that the cell is that the cell wants to start moving, right? That the cell wants to enter a proliferative migrating phenotype. And at the same time, you have basically a decrease in the activity of all of the other things that actually do the opposite. So the cell becomes activated, and what you get is the sprouting of those endothelial cells and the initiation of angiogenesis, right? So again, take-home message here, you basically have signals that promote quiescence and they stabilize the endothelial cell. And there are signals, primarily collagens, that promote angiogenesis. Now, angiogenesis is a really complicated process. So there are several types of receptors, including ephrin receptors. Ephrins are the signaling molecules, and they bind to ephrin receptors or F receptors. Angiopoietin is sort of a kind of a growth factor, binds to Ti receptors. There's delta notch, and I mentioned delta and notch in the signaling lecture, but all I really want is for you to remember that delta and notch play a role here. And then the most important factors are the vascular endothelial growth factors, or VEGFs, right? VEGFs bind to VEGF receptors. And this figure basically shows the different types of VEGF receptors that are known. These VEGF receptors bind 5 different VEGF molecules, right? VEGF A, B, C, D, and placental-derived growth factor. Those are the 5 vascular endothelial growth factors. And they differ in their binding affinities for the different receptors. Not all receptors bind all of the VEGFs. And I don't necessarily want you to remember or memorize the specific pairings, for instance, because there's the receptors can homodimerize, they can heterodimerize, there are co-receptors. It's very complicated. So I don't want you to focus on the specifics of, VEGF A binds to VEGF receptor 2, blah, 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 right? I don't want you to memorize any of this. I do want you to appreciate that this is a very complex process, and the different pairings ultimately have different outcomes. And these receptors are actually involved both in angiogenesis and vasculogenesis. So both of the processes that I mentioned a little earlier. Now, the VEGF signaling processes are very, very complex, as you can tell from this image. I'm going to try to walk you through it, and what you'll see is that there are certain things that are color-coded. The things that I think are necessary, the things that I think are important for you to sort of pay attention, I'm going to emphasize. But the take-home message is the big picture here. So in this particular image, what you have is a single VEGF receptor, which is VEGF receptor 2, right? With its extracellular domain. And VEGF receptor 2 is known to very importantly bind VEGF A, C, and D. VEGF A is sort of the standard VEGF. Here, inside of the cell, you have the intracellular domain of the receptors. VEGF receptors are of the tyrosine kinase receptor family, right? And you remember that receptor tyrosine kinases have the ability to cause cross self-phosphorylation, right? That's sort of their activating pathway. And all of these Y with numbers represent tyrosine residues in the intracellular domain of the receptor that may or may not be phosphorylated as a result of receptor activation. So that's sort of the center column here, right? The receptor and the different tyrosines. Now, these tyrosines that I just highlighted there, 801, 1054, and 1059, are probably the best characterized. So when people study these, what people look for is the phosphorylation of those tyrosines. Now, remember that the way that tyrosine kinase receptors become a signal is the phosphorylation causes or creates binding sites for SH2 molecules, right? So the receptor, the intracellular domain of the receptor turns into this sort of signaling hub, right? 1175 is also important. And the reason why is because the phosphorylation of 1175 creates this SH2 binding domain where you get GRAP2 and SOS, but also SRC and SHB. Again, don't focus on the specific numbers. I'm just trying to correlate what you see here with things that I've said before. Now, everything that's highlighted in blue, so the activation of SOS, activation of phospholipase B, phospholipase C beta 3, and then the activation of focal adhesion kinase, IQ-GAP1, and all the MAP kinase cascade, all of that stuff is usually associated with migration. So activation of the receptor and signaling via these pathways triggers migration because, again, actin polymerization, turnover of focal adhesions, remodeling of cell-cell contacts, all of that stuff is associated with migration. Now, 1175, by the activation of phospholipase C gamma, triggers IP3, increases in calcium, and the activation of the RAS pathway, which ultimately leads to changes in gene transcription, gene expression, and that changes the phenotype of the cell towards a proliferative state, right? So ultimately, the outcomes of these gene transcription lead to increases in cyclin expression, entry into the cell cycle, or re-entry into the cell cycle, and cell division. Some of these pathways are actually associated with inactivation of caspases, right? If you inactivate caspases, then you're basically preventing those cells from dying, and that's a survival sign, right? And finally, and perhaps what I care about the most, is that there is the activation of the SRC kinase and the activation of a number of other proteins that lead to the internalization of weak adherin. Weak adherin is the most important intercellular junction molecule in endothelial cells. So if you internalize this weak adherin, you're basically removing it from intercellular junctions, and you're basically losing that contact inhibition, right? At the same time, this pathway leads to the production of the activation of nitric oxide production and the increase in vascular permeability, right? And permeability, so increased vascular permeability, enhanced cell survival, acquisition of a migratory phenotype, and a re-entry into the cell cycle gives you angiogenesis, right? All of those four factors are basically hallmarks of an angiogenic process. Proliferation, so that means re-entry to the cell cycle. Cytoskeletal remodeling leading to migration. Inhibition of pro-apoptotic signals leading to cell survival, and increases in vascular permeability are the hallmarks of angiogenesis, right? Any questions about this? Okay. So why do we care about angiogenesis? For a number of different reasons. But perhaps the most important one is that it's critical for tumor growth, right? So when you have tumor cells and they start forming a malignant growth, these tumor cells will produce a primary tumor. And as these cells grow, right, as the tumor grows, I want you to sort of go back to the second slide today where I told you, that plant organism that is a multicellular organism. There's plenty of oxygen in the outer layer, but the deeper you go, the less oxygen you get. And that means that in order to support tumor growth, particularly of those tumor cells that are buried deep into the tumor, those cells need oxygen too, right? So as the tumor grows, the core of the tumor becomes more and more hypoxic. And when the cells become hypoxic, they start making angiogenic factors. Those angiogenic factors diffuse away from the tumor, and they bind to receptors in the surrounding blood vessels, and they induce angiogenesis. So angiogenesis is necessary for tumor growth, right? In other words, the tumor tries to get its own supply of blood so that it can continue growing. Now, the more angiogenesis you get, the more fuel you have to promote the tumor growth. The more a tumor growth, the more the tumor grows, the more it changes its phenotype. And at some point, a tumor may acquire an invasive phenotype so that the tumor cells become migratory, and they basically make their way to the blood vessel. They enter the circulation, and then they cause metastasis. And when the tumor cells extravasate, they begin proliferating in a secondary site, and some of those cells are going to proliferate right away. Some others are going to enter a state of dormancy, and then they're going to be reactivated later down the line. That's what happens when people go into remission, and then the cancer reappears years later, right? Though there were those cancer cells that actually metastasized, but they were dormant for quite some time. So that's basically, that's the importance of angiogenesis in terms of cancer, right? But we do have to talk a little bit about the endothelium as a barrier function. I told you that one of the things about angiogenesis was that vascular permeability had to increase, right? So in cancer, the endothelium of those blood vessels that are close to the tumor actually look nothing like the normal endothelium, right? So in cancer, the endothelial cells are sprouting sort of constantly, and those areas that are highlighted in red basically are places where the cells are not really touching each other, right? And those are areas where the endothelial barrier is lost, right? And over the course of months to years, the pro-angiogenic factors basically increase the permeability of the endothelium, particularly close to the tumor. And there's a transient increase in leakage. Again, transient, we're talking about months to years. But at some point, the leakage sort of stops. And there are a number of different things there are a number of different reasons that explain why the leakage itself goes away during sort of like in time. But the important thing is that the permeability of this barrier remains elevated because you have all of those gaps between the endothelial cells. So it shouldn't surprise you that VEGF signaling is actually the target of many very important anti-cancer chemotherapeutic drugs. So it's a very, very common target for the treatment of solid tumors. And this is a non-exhaustive list. There are some of them that are monoclonal anti-VEGF antibodies. So they basically, this antibody sequesters the signal so that the signal can't act on the receptor. There are there's a recombinant VEGF protein that basically mimics VEGF and prevents, it mimics VEGF and it binds to the receptors, but it doesn't cause activation of the signaling pathway. And then there are antibodies against the receptor, and then there are tyrosine kinase inhibitors. This is just an FYI slide. I only really need you to understand that targeting the pathway is used therapeutically in the fight against cancers. These are relatively efficacious drugs, but none of them provide a complete cure, so we have to keep doing research and asking for money to do more research to improve these anti-angiogenic therapies that prevent cancer. Now, the thing is that there are other pathological situations, such as ischemic disorders, where you kind of want the VEGF activity, right? And in fact, VEGF gene therapy was trialed for critical limb ischemia, but it didn't work out. So again, there are pros and cons to using the pathway for this for therapeutic purposes, and with some things have worked out, some other things have not. Finally, I wanted to tell you a little bit about the endothelial barrier, and I want to tell you about the endothelial barrier because this is what I do for research. This is the thing that's really close to my heart. So on the top panel, you have the normal endothelial barrier that's sort of tight, permeability is low, leakage is low. In acute inflammatory scenarios, so when you have, this is what happens when you get a mosquito bite or when you bang your head against your cupboard in the kitchen, that anything that causes a swelling, right, will cause an acute inflammatory response. And in the acute inflammatory response, the endothelium becomes sort of open. So you see that the intercellular junctions are transiently open, and that causes an increase in permeability and an increase in leakage, but this lasts for a very limited period of time, right? We're talking about minutes. In the worst case scenario, it lasts a day or two. And this is something that's mediated exclusively by endothelium in the postcapillary venules, and I'm gonna tell you why. Now, in situations where the presence of pro-inflammatory agents is chronic, so when these things last for a long period of time, you end up having a vessel enlargement. You see that this endothelium is a little smaller than this one. And then there's a chronic increase in permeability, and then there's chronic leakage. And this is what happens in some autoimmune disorders. This is sort of people consider cancer as an inflammatory condition. So this chronic inflammation sort of precedes the type of inflammation that you see in cancer patients. So the endothelial barrier, right, in tight endothelia, is formed by several different types of proteins. We can classify these proteins in tight junction proteins and adherence junction proteins. The tight junctions, which are mediated by claudins, occludins, and junctional adhesion molecules, or JAMs, all of them dock to the actin cytoskeleton via ZO, or zonula occludins molecules. And these are the proteins that are really, really, really important in areas in endothelia that have the tightest of barriers, right? So these are very highly expressed, for instance, in the endothelium of the brain, right? And also in the endothelium of most arteries and arterioles. All of those endothelial cells also express adherence junctions, and they're mostly mediated by vascular endothelial cadherin, or VE cadherin. And VE cadherin is anchored to the actin cytoskeleton via a complex of catenins, basically, alpha, beta, and P120. And then there's also nectin and ephedrin, but I don't really care about those. In endothelia with intact barriers, you have a really good overlap of the tight junction and the adherence junctions so that the contact between two endothelial cells is very good. But when there are pro-inflammatory signals that lead to barrier disruption, usually what happens is that the adherence junctions are disassembled and the tight junctions remain in place, right? And that basically helps the two cells to remain attached, but to also increase the permeability, right? Now, I want you to think about this a little later. In post-capillary venules, the tight junction proteins are usually absent. And that means that the only proteins that are really there are the adherence junctions. So what I'm going to show you is actual experimental data, not generated by me, but generated by colleagues of people that I know that have kindly provided these pictures for us. So this picture shows the microcirculation in the pouch, in the cheek pouch of a hamster, right? Tiny little things that, you know, fill up their cheek pouches with stuff. The cheek pouch is a really great experimental model for all of this because it's easily accessible, and then you put all of these things under the microscope and you get this. So this hamster was injected with a fluorescent molecule called phytidextrin. This is what we call a high molecular weight dextrin. Basically, this dextrin mimics the proteins that are present in the plasma. And the proteins that are present in the plasma normally stay in the plasma, right? They don't really leak because they're too big. And what you can observe is there is this central vessel here. This is probably an arterial, if not a vein. Yeah, this is an arterial. And in the background, over there, you have venules and arterials, like tiny. And those are mostly post-capillary venules. And in the background, the tiniest things that look really faint, those are capillaries, right? So this is the control situation. Nothing has been, we haven't really stimulated with anything. But then what we're going to do is we're going to put a relatively high dose of histamine, right? The same molecule that mediates our allergic reactions and all that stuff. That is actually a very potent pro-inflammatory agent. And when histamine binds to endothelial cells, it causes nitric oxide production. I'm going to tell you a little bit about that later. So when you put histamine, that happens. What you're seeing, first of all, take a look at the central arterial. It dilated, right? That's the effect of nitric oxide in the arterial. Most of the vessels that actually dilated, they seem to be arterial side. Now, you see all of these foci where the green becomes more intense, right? Those are what we call leakage sites, right? That and you can see that the leakage is sort of diffusing away. It's sort of diffuse in its appearance. That is the fluorescent dextrin escaping from the blood vessels into the interstitium, right? And something that's, I think, important for you to understand is that those foci are only located in post-capillary venules. The sites of leakage only occur in post-capillary venules, right? So this thing that you see with histamine can be replicated with mediators like bradykinin, histamine, platelet activating factor, and also VEGF, right? All of those molecules will increase endothelial permeability, right? And they only really act on postcapillary venules, where the barrier is only really made of adherence junctions. Does that make sense so far? Any questions about this? All right. So now, bradykinin, histamine, the platelet activating factor, vascular endothelial growth factor, all of those have different receptors. But what they have in common is that all of them converge in activation of eNOS and nitric oxide production, right? So what happens is that, well, in this particular figure, what you have is endothelial cells in culture, and that have been stained to show vascular endothelial cadherin, right? And you see red, that's the cadherin molecules forming the barrier, right? DAPI in blue, that shows you that that's the nucleus of the cell. And the tiny little things in yellow represent sites of leakage. That's where the barrier has been lost, and you can actually, with some experimental manipulation, you can sort of see what's under the cell. When you expose these cells to VEGF-A for 2 minutes, you see that there's much more leakage now, because the cells, there is less cadherin forming junctions. The cells basically contract, and the sites of leakage increase in size. And this happens because, again, there is production of nitric oxide by eNOS, and activate the concurrent activation of several different signaling pathways that ultimately lead to the internalization of V cadherin, right? So V cadherin becomes internalized by endocytosis, right? And endocytosed V cadherin cannot form the barrier anymore, right? The details of this are not super important. A lot of research has been done on the ways to regulate V cadherin in particular. Several signals are correlated with V cadherin internalization and barrier disruption, including the phosphorylation of several residues here and the nitrosylation of some of these catenins. Now, what is the role of nitric oxide here? And this is actually kind of interesting, and it's very cell biology forward. So this data that I'm showing you here shows data that was generated in 2 different animals, 2 different mice. The mice on top is the wild type mouse. So that's a normal, healthy mouse that has all of the genes. The mouse on the bottom is a mouse that doesn't have the gene that encodes for eNOS, and therefore the endothelium of that mouse cannot produce nitric oxide, right? So what these people did here was to basically they threw a pro-inflammatory agent. In that case, if I remember correctly, is PAF, platelet activating factor. And as you can see in the wild type mouse, PAF increases leakage significantly, and over time that leakage basically decreases. And the signal decreases for several different reasons. In part because the barrier over time gets restored, in part because the lymphatic system basically picks up the fluorescent molecule and basically takes it away. However, in the eNOS knockout mouse, right? So the mouse that doesn't have eNOS, PAF doesn't have the same effect, right? You see that leakage increases very, very minimally, but it's nothing like the wild type. And quantification of this looks even more dramatic. In the closed circles, you have the normal wild type mouse, where permeability increases rapidly and then decreases over time. And in the knockout mouse, basically you don't have that. This data suggests that nitric oxide production by eNOS is necessary for this increase in vascular permeability. Several pieces of data indicate that one of the key aspects of this process is not just nitric oxide production, but delivery of this nitric oxide to specific regions of the cell. So in the bottom panels, what you have is endothelial cells in culture that have been stained for enos. And you see enos in green. And under control conditions, a significant portion of enos is actually found on the cell borders, on the cell membrane, right? When you stimulate these cells with a pro-inflammatory mediator such as PAF for 3 minutes, enos disappears from the membrane, right? You don't really have that very nicely delineated localized enos on the membrane, right? So this basically tells you that a pro-inflammatory agent is basically causing the trafficking or the movement of the enzyme. Usually, and for different reasons, we know that this happens, the enzyme is internalized into the cytosol of the cell. Now, here what I'm trying to highlight is the places of the cell where you do have enos in the control versus the places where it's membrane bound in the treated cells. You see that there's a lot less enos in the membrane. However, when we stimulate, in this case, this is data from an animal. When you stimulate the when you cause increased permeability with PAF, but then basically block that with this molecule called ATPT. ATPT is a cyclic AMP analog. So it basically does some of the things that cyclic AMP does normally in the cell. And you can see that, in this particular graph, the closed circles still represent PAF alone. So permeability increases and then decreases, just like in the previous experiment. But when you put ATPT on the animal, right after putting PAF, the permeability response is blunted, right? In other words, this molecule here is basically stopping or ameliorating the this state of hyperpermeability. And at the cellular level, what we know is that cyclic AMP acts via EPAC1. So this molecule here, ATPT, is an activator of EPAC1. And EPAC1 causes a number of different things that lead to basically the remodeling of actin cytoskeleton and the insertion of V cadherin on the cell junctions, right? Why is this important? Because the more V cadherin you have in the cell junctions, the better your barrier is, right? And this means that if you have a pro-inflammatory agent, right, if you stimulate cyclic AMP signals, then you can sort of counteract that pro-inflammatory state. And when we did this experiment on the cells, again, we stimulated the cells first with PAF, and then we put ATPT, enos went back to the membrane, right? You can see that there is enos in the cell junctions of the cells. Maybe not to the extent of the control situation, but certainly more than in the PAF, in the cells stimulated with PAF alone. What this is telling us is that the strengthening of the barrier depends on cyclic AMP causing the movement of enos back to the membrane. And together, this tells us a very interesting story that it's the location of nitric oxide production that determines permeability. And what we tried to publish, and well, we actually did publish this back in 2023, is that agonists that cause inflammation trigger internalization of enos and cytosolic nitric oxide production. And this production of nitric oxide in the cytosol is associated with increased inflammatory hyperpermeability. But then there is a really cool delayed increase in cyclic AMP that I didn't really tell you about this because we don't have time. But ultimately, there's this weird and still unexplained delayed increases in cyclic AMP that leads to EPAC activation. And somehow, we don't really know why yet, activation of EPAC causes eNOS to move back towards the membrane. And it's the movement to the membrane that causes the termination or the inactivation of this inflammatory hyperpermeability. And what we think is that this is an interesting and potentially therapeutic avenue to treat important situations such as compartment syndrome. That's usually a really serious complication of limb reattachment surgery. So when you have a patient that suffers really terrible trauma and you have to reattach a finger or a hand or an arm, the detached limb has been ischemic for quite some time, right? Because it's detached from the body, and therefore there's no supply of oxygen. And it turns out that when you reattach it and you reestablish blood flow, that reestablishment of blood flow, called reperfusion, causes a massive inflammatory response. And that massive inflammatory response can actually lead to tissue necrosis because the damage is really serious. So if you could actually figure out a way to stimulate these pathways that prevent hyperpermeability before you reattach the limb, or before you, for instance, in organ transplant surgery, in those circumstances, what you would expect is to minimize the damage that this reperfusion can cause in the tissue, and that should, or we hypothesize that it should improve the outcomes of these procedures, right? And that's it. Any questions? All right. Oh, wait, there's something in the chat. Maybe that's... Ah, thank you. I appreciate it. Thank you all. If you have questions, please feel free to email me. I will tell you right away, I will be away on vacation next week. So if you email me if you email me next week, I'm probably not going to get back to you right away. But email me nonetheless. I'll get back to you when I get back. All right. Take care and thank you.